Hello and welcome to this lecture. My name is Leo Kirug, and I'm really happy to present this talk, although I must say that it's a very difficult topic for various reasons. Some of them will be discussed during the lecture, but at the same time it's also the topic that is the most interesting and close to my heart at the moment in my life. I'm very interested in the questions of cognition, consciousness, and the neural substrates, neural correlates of consciousness and cognition, the ways in which we learn about the world, the ways in which we project our expectations and emotions onto our picture of the world. Some of this will be discussed in the lecture. And at this point I will switch to audio only and hope that you enjoy the show. In part one of this lecture, we shall mostly focus on the neural representations of personal space, of extrapersonal near space, and also of mental processes. This lecture course is based primarily on the book of Kandel, the 5th edition, and I'm covering here chapter 17, From Neural Cells to Cognition. And here you can see the table of content of this particular chapter. It is heavily focusing on the neural representations of mental processes, and it also talks about the modified representation that can be modified by experience. The scientific method as a whole is based on a number of important assumptions. One assumption is that there is an objective, physical, real world out there, outside of the observer, and that there is an observer that is separate from the environment, from the real objective world, and that there is a process that we call cognition that is an objective representation of the objective world, of the object. This general framework assumes that, of course, the world is not accurately and completely represented in the mind of the observer. But the assumption is that the representation that we have in our minds is at least partially correspondent to the features of the world, although it is always from a single perspective. It's like a point of view that gives you only one projection of a three-dimensional object onto a two-dimensional space, figuratively speaking. There is also a growing recognition that in addition to the world out there, what we perceive in our minds is a sort of a playback of a closed-loop camera recording, which includes the memories, the prior knowledge, our predictions, and we'll talk more about the predictions in the course of this lecture, and the assumptions or educated guesses that we make. These relatively new discoveries about the predictive properties of our mind and the overlap between predictions and the input, the sensory input, is very interesting, but we'll talk more about this in the second part. For now, let's focus on the representations. Perhaps one more thing to be added about the representations of the external world, as well as our own mental activity, is that it's heavily colored, flavored, and distorted by our emotions, by the level of our attention, and by the kind of environmental noises and distractions that we get. And this is all important to keep in mind when we talk about the accuracy and the precision of the representation. Let us use the visual system as the best studied sensory system that we have, and also arguably the most important one for us humans, because we so heavily rely on our vision, and because our vision is binocular, three-dimensional, colored in such a way that it makes it quite comfortable for us to use vision to orient ourselves in the environment, as opposed to for example, smells or tactile information. As illustrated in this slide, you can see that the visual representation of the external world bears many similarities to the diagram that I drew on the left, the camera obscura or the photographic camera or video camera for that matter. There is the world out there, which then the reflected light or emitted light is perceived by our photoreceptors in the retina. The information is then transmitted to the visual cortex, and the picture is reconstructed in the visual cortex as a particular perspective, a particular two-dimensional projection, a snapshot in time of the objective external reality. The similarity between the photographic snapshot and the visual representation of the external world by our visual system is striking, and it's supported by a great deal of mechanistic information, mechanistic knowledge that we have gained by using the reductionist approaches that I will talk about later in the course of this lecture. So we know quite precisely what kinds of cells, what types of cells, how many different types of cells in which sequence, etc., participate in relaying this information through their synaptic connections all the way from the receptor cells in the retina to the higher processing centers in the visual cortex and beyond. How do we as scientists, as professional researchers, gain this mechanistic detailed information about our visual system and other parts of our brain, other parts of our bodies, and about nature in general? Well, the main tool of science is reduction. The reductionist approach consists in splitting things up into more and more detailed subparts in an ultimate quest for finding the building blocks of the universe. What is everything made of? And of course it used to be the atom. The atom itself means in Greek the indivisible. It's like individual. Indivisible, indivisible, unsplittable. Of course, the atom was later split up, and more elementary particles were discovered and are being discovered, and then under that there are strings and more supposedly elementary and undivisible building blocks of the universe. And it's an incredibly useful tool in terms of practical applications of science, because the more details we get, the more we're capable of reconstructing things and building devices and apparatus that are useful in everyday life. This is how science is very helpful through engineering and entrepreneurship and businesses in making people's life easier. So how does reductionism work? Well, consider we are looking at the starry sky during the night, and we're interested in one object, one particular object in that sky, the moon. We can just observe it with our naked eye, but it becomes more interesting if we make a tool, such as a telescope, just a collection of lenses, the glass lenses. And then we look selectively at this particular object and we ignore everything else. And this is also a fundamental property of scientific research and of knowledge in general, cognition in general, is that when we consciously consider something, we ignore everything else. We separate it from everything else, separate the figure from the background. So now the figure is separated. We're ignoring the light from all the stars in the sky, but we are looking at this satellite, the small planet that is rotating around the Earth, and we can zoom in ever more and discover more and more details. We can finally build a map. We can build a mental representation through our cognition and through the equipment that we use, of the moon as an object. In a very similar way, and on the micro scale, the neuroscience, for example, or other micro scale branches of science, are building the mental maps, mental representation maps of what the brain is made of. So we can observe the brain in biopsy, or autopsy rather, as a macro object with our naked eye, but then if we put it under the microscope, which is just an inverse of the telescope, pretty much, and we use special methods of contrast enhancement, such as overexpression of fluorescent proteins of different colors, or Golgi staining, etc., we can see the shapes of individual parts of the brain, the neurons, the astrocytes, the microglial cells, the vessels, all sorts of other cells that we discover in the brain. And then by using more advanced tools and time-lapse imaging, the scanning, laser scanning microscopes, some of which I'll be mentioning in the third and fourth part of the talk, and using the tools such as calcium-sensitive fluorescent proteins, we can also look at the functional readouts. We can visualize the signaling between neural cells in real time in the brain of a living animal. And that also allows us to build maps, such as the connectome, the connections between different neurons, different parts of the brain, different regions over time in space, etc. And it's very important to keep in mind that reductionist is just one direction in which we go to refine to get the details, but we always need to come back to holistic, to the whole, to the holistic part of this duality, reductionism versus holistic, and reconsider the moon as a whole, or the galaxy as a whole, or the brain as a whole, or society, and the species, the human Homo sapiens species, etc. So contextualizing the reductionist knowledge, detailed mechanistic knowledge, is an important step. Otherwise we get buried into the details, and as Einstein said, many scientists do not see the forest, they only focus on the trees, and they forget about the big picture, and that is damaging for science just as much as lacking the tools for reductionist analysis of the details. And to follow further the advice of Albert Einstein, when he said that it's important to see the forest and not just the trees, he suggested that it's very useful to have in mind the historic perspective, the history of the development of our tools as they get more and more refined and discover more and more of details for us, and the importance of bringing it back into the holistic context. So this is a complicated, busy figure. We're still working on it. This will be part of the editorial in a special issue of Cell Calcium that we are now collecting papers for from different authors working in living animals, living behaving animals using microscopy and electrophysiology. But this figure is intended to draw a picture of the history of neurosciences from the early days of neuroanatomy before any of the microscopic or other techniques were available, just by observation of the human organs, the functional neuroanatomy, the studies of Galen in the beginning of the first century. He was looking at the wounded gladiators, actually, and also traumatized animals to see which parts of their brain were responsible for what kind of functions. Then, of course, later on, the development of microscopy has become incredibly useful in moving science more and more towards the reductionist and detailed analysis. And So just to explain, this tree has two branches, and one is the studies in humans, in Homo, and the left one is the studies in animals, in Bestia. And the deviation from the center of the trunk of the tree represents the increase in reductionism and decrease in the holism. So here are some abbreviations. The extreme reductionist approach is in vitro, when you grow cells, neuron cells, on glass and study them in separation from each other. The ex vivo preparation, when you cut the slices and record electrophysiologically from them. In vivo is in the living animal, but perhaps anesthetized. And finally, in vigilo is the term that I'm trying to promote, I coined. It means in the vigilant, in awake animal, and same applies also for humans. So what you can see, the tendency here is that for 2,000 years or so, there was a trend towards more and more in vitro, more and more reductionist methods. But lately what we observe is, for example, with the development of non-invasive techniques, such as electroencephalography or MRI and positron emission tomography imaging that can be used both on animals and on humans, and then also microscopic imaging of calcium signaling and stimulation of individual cells or cells ensembles with optogenetic tools, etc. And also the use of the stem cells that can be cultivated in vitro and then used in humans in clinical trials. So the trend is that the reductionist methods are now being recontextualized and brought into the whole body, the whole animal, the whole human context. And to me personally, it seems that this is the most exciting time in neurosciences, because now we can discover mechanistic details using reductionist methods in the context of a whole animal combined with behavior. And this is what I will be showing to you in the third and the fourth part in talking about my groups and the core facilities approaches that we use there.